All right, so this screencast is dealing with vision. Our objective here is to define transduction and to look at how we transfer our visual system into neural impulses and how our brain interprets them. So transduction is the conversion of one form of energy into another. And in vision, it's taking the stimulus energy of electromagnetic waves and sending them in, or turning them into neural impulses where our brain interprets them in our visual cortex. What you see here is the spectrum of electromagnetic energy. You can see that the part that's visible to the human eye is actually quite small. The spectrum ranges from gamma rays, as short as the diameter of an atom, to radio waves that can extend over a mile long. Other organisms are sensitive to different portions of the spectrum. For example, bees cannot see red, but they can see ultraviolet light. When we look at the process of vision, there are really two steps. First one is gathering the light. Light is reflected off of objects and is gathered and collected by our eye. When we look at color, it really depends on two factors. This is a really good graph to help you understand and process how we perceive color. The first is on the left-hand side, and it's talking about wavelengths. Wavelengths is the distance from one wave peak to the next, and that wavelength will determine the color that we perceive, or the hue. So when you look at the blue electromagnetic waves, the peaks are very close to one another. It's a very short wavelength. And the shorter the wavelength, the more bluish colors we perceive on the rainbow, bluish and purple. The longer the wavelength, like you see with the wet red, the distance from one peak to the next is extended, and we see reddish colors. So when you think about red, orange, yellow, green, blue, purple, the longer the wavelength, the longer the reddish colors you see. The shorter the wavelength, the more bluish colors you see. The other part that is determined by wavelengths, or I shouldn't say wavelengths, the other thing that's determined by the type of color that we see and its intensity is determined by amplitude. And amplitude is the height of the wave. You can see from the top of the wave on the right-hand side of the chart to the bottom valley of the wave. So the greater the amplitude, the brighter the color. The smaller the amplitude, which you can see determined by the light green at the bottom right-hand corner, the duller or softer the color. Step 2 is within the eye. We're going to take a look at the structures within the eye and look how the process of transduction works. The cornea is your protective covering. This is where the light enters. We have corneas to keep out flecks and particles and dust, and you know how sensitive the corneas can be. If you've ever had a cornea scratch or a cornea tear, it's been a pretty miserable experience. So we know that the cornea is very sensitive. It protects the eye. It actually bends the light to provide focus. The next is the adjustable opening. It's the black portion of the eye. The pupil is really under the control of the iris. The iris is the colored portion of your eye, and that muscle allows for the pupil to either dilate, which is what you see in the picture right here, or constrict, very small, depending on how much light is entering the eye. When it's really dark, we know our pupils dilate. When it's really bright, our pupils constrict. So the iris that you see right here adjusts light intake. It dilates and constricts in response to the light intensity and even to other things like hormones, inner emotions, and that sort of thing. So the iris really does control the pupil. And this is the colored portion. It's the muscle. Our lens is very curved and flexible. Another name for the lens flexibility is accommodation. You want to have this flexibility in your eyes because it allows for your eyes to focus. It's the lens that actually flips the image upside down while your brain in your visual cortex in your occipital lobe actually flips it back. So you can actually feel the lens change shape or accommodate when you look at far away objects and then look at close-up objects. You can actually feel the change in the lens when it's focusing. Your retina is the inner surface of your eye. It contains the receptor rods and cones. Rods are responsible for us perceiving images in the dark because it helps us with gray and black, where cones help us perceive color. It's important that you know their function. Rods, black and gray, cones, color. And it also contains layers of neurons that allow for the processing of visual information to start. This is a nice visual to show you how the candle is actually flipped upside down by the lens near the point of central focus, or the fovea. You can also see where the optic nerve leaves the eye and sends the information to the thalamus. Acuity is our sharpness of vision, and it can change by small distortions in the shape of your eye. And it can be affected by small distortions in the eye's shape. So what you see with normal vision are rays of light converging on the retina of a normal eye. This occurs with both nearby objects and with appropriate adjustments in the curvature of the lens for faraway objects. In the middle you see nearsighted vision. And if you take a look at the candle, you can see how in the person with nearsighted vision, the light rays from distant objects focus in the front of the retina. And when their image reaches the retina, the rays are spreading out and it actually blurs the image. And if you're really nearsighted, you actually see nothing clearly. Glasses, contacts, laser surgery can reshape the cornea and can actually correct the problem. If you're farsighted, light rays from the nearby objects reach the retina before they have produced a focused image, making nearby objects really blurry. In children, the eye's ability to accommodate usually makes up for this problem. That's why children rarely need glasses. But they may suffer from eye strain, from overusing the eye muscles, and sometimes you can see kids getting headaches, and you check their vision, and oftentimes that problem is corrected. This next slide is a really good slide for you to memorize in terms of transduction. If you followed a single particle of light energy into your eye, you would see that it first makes its way through the cornea, through the pupil and iris and lens, and then it hits the back of the retina. So this slide actually shows you a cross-section of the back of the retina. And the light triggers the rods and cones to be stimulated. And that starts the process of neural transduction. So if you follow the arrows, the first thing that is stimulated is the bipolar cells. The bipolar cells trigger action potential in the ganglion cells. The ganglion cells' axons, you can see, are really long. They converge to form the optic nerve. The optic nerve leaves the eye en route to the thalamus, which then transmits information to the visual cortex in the opposite hemisphere. So you need to know, and it's nice because it is in alphabetical order, bipolar cells, ganglion cells, optic nerve, thalamus, and visual cortex. That will be on the test, and you have to know that order and sequence. The blind spot that we all have is the point where the optic nerve leaves the eye. It creates a blind spot because there aren't any receptor cells located there. But we don't notice blind spots because we have two eyes. And what's in one eye's blind spot, the other eye picks up in their field of vision. This is a little fun activity to test and see where your blind spot is. You can cover up one eye, focus on the dot, and when you move the book or the PowerPoint away at a distance, the car actually disappears. Magical. This is a nice illustration just showing you how information that leaves the eye via the optic nerve crosses over the corpus callosum, and it is relayed via the thalamus to the visual cortex in your occipital lobe. Blind sight is a type of disorder where a person actually has blindness in part of their visual field because of the visual cortex being destroyed, possibly from a stroke or a lesion from surgery or just some kind of brain injury. And so if you get a chance, watch the YouTube video. It gives you a better understanding of how this works. But they're actually blind in part of their visual field because of damage to the visual cortex. There are two theories that you have to be familiar with that help us understand how we perceive color. The first one is called the Young-Helmholtz trichromatic theory. The idea is that our retina contains three different color receptors, cones. One is red, one is green, and one is blue. And when light hits the retina and stimulates these cones in combination, they can produce the perception of any color. So you see the mixing paint, that palette, it's actually known as subtractive color mixing because it subtracts wavelengths. So mixing all three colors leaves you with black. Mixing light is additive because wavelengths from each light in the mix reach the eye, and mixing all three primary light colors creates white. So those who are color deficient, they lack functioning red or green sensitive cones, or sometimes both. Their vision seems normal as monochromatic instead of trichromatic. Another theory that helps us understand the color that we perceive is the opponent process theory. This is a really good, once again, if you get a chance, click on this link and it shows you how this theory works. But it's defined as a theory where opposing retinal processes, red opposing green, yellow opposing blue, white opposing black, enable color vision. So for example, if you stare at the dot long enough on this particular flag and look away at a white surface or even close your eyes, you'll actually see the colors of the American flag. You do need to know which colors oppose one another: red, green, yellow, blue, white, and black. A person with color deficiency, which is more typical in men than women, about 1 in 50 people are color deficient. If they have red-green color deficiency, they would not be able to perceive the 74 within this circle. The last slide is called color constancy. It's when we perceive familiar objects as having consistent color, even if the illumination changes and it alters the wavelengths reflected by light. What really seems kind of almost trippy a little bit is the fact that the three blue dots in this cube, they're actually all the same color. But because the one that has what looks like yellow dots next to it is so dark, it almost looks like a neon blue. The top one that has the most illumination almost looks navy, where the one on the side almost is like a royal blue, somewhere in between. They look like different colors, and you have to just believe me on this one, but if you were to put all three of these dots next to each other, they would be the exact same blue. So what it means is that our surroundings affect the color we perceive, but the color actually does remain constant. And that is the last slide on the visual sensation screencast.